Assalamualaikum and a very good afternoon to you all. I am Lieutenant Colonel Dr. Sara Waqar Khan, a consultant immunologist and assistant professor of pathology, currently serving at CMH Peshawar. Today, now we are heading towards the last session of our immunology conference, which is immunodiagnostics, which is the backbone of immunology. There is a constant evolution and advancement in this field. This afternoon, our astute speakers will share some of their amazing work in their respective fields. First, I would like to invite the chair of the session, Dr. Nadeem Afzal, who is Professor of Immunology and Pathology, Saeed Akhtar Medical College, Lahore, and Consultant Pathologist at Farooq Hospital, DHA, Lahore. He is a graduate of Allama Iqbal Medical College in 1986. Started his career from NIH Islamabad, did his MSc Medical Immunology from University of London, followed by PhD, did CMT 2 years ago, and trained and supervised over 50 students in immunology. He has over 90 publications. I would like to call upon Dr. Nadeem Afzal. Please welcome him as chair of the session. The second chair of the session is Lieutenant Colonel Dr. Hamid Nawaz Tiku, who is currently Associate Professor of Pathology and Consultant Immunologist at CMH Lahore. He is a graduate of Army Medical College, 20th batch, did his FCPS in Immunology from FIT Rawalpindi in 2010. His special areas of interest are immunogenetics and immunoinformatics, on which he has over 55 publications. So please come to the stage. Now we'll formally start this session.
Bismillahirrahmanirrahim. Thank you very much for the nice introduction. After knowing that I was graduated in 1986, I was thinking of that, oh, it's way back. I request Dr. Major Chipu to please, if he can begin the proceedings. Thank you.
Thank you, sir. Our first speaker today is Dr. Sabahat Sarfraz. She is Assistant Professor of Pathology at Dow University of Health Sciences. She has multiple publications on clinical immunology and she is serving as Chief Immunologist who started renal transplant program at Dow University. She has commendable work in immune deficiency at Dow University. She will talk about SSA antibodies if these are nephrotoxic. Dr. Sabahat.
Thank you, sir, for your kind introduction. Bismillahirrahmanirrahim. Assalamu alaikum. Respected chairs, seniors, dear colleagues, juniors. Dr. Sabahat Sarfraz, serving Dow University of Health Sciences as Assistant Professor. My talk title is Are Anti-SSA Antibodies Nephrotoxic? I'll talk about autoimmunity, what are anti-SSA antibodies, a literature review, and will share my study results. Autoimmunity, we all know, is basically a self-damaging immune effector response which manifests in various autoimmune disorders. The word horror autotoxicus was first coined by Paul Ehrlich in 20th century to depict the disastrous consequences of this condition. Anti-nuclear antibodies, now called as anti-cellular antibodies, are the defining features of autoimmunity. Basically, they are the first screening tool which helps to clinically identify the impaired tolerance which manifests as autoimmunity. Followed by the positivity of these antibodies, antigen-specific antibodies are assessed in order to differentiate between different autoimmune disorders. Here you can see there is considerable overlap between the extractable nuclear antibodies and the associated autoimmune disorders. Anti-SSA antibodies are the most frequently detected anti-SSA antibodies. Previously, they were found only in patients with Sjogren's syndrome and systemic lupus erythematosus. But recent researchers have mentioned their association with many other rheumatic as well as non-rheumatic disorders. Despite the presence of many new autoantibodies, anti-SSA antibodies is the only antibody which is included in the American College of Rheumatology diagnostic criteria for Sjogren's syndrome. Clinical importance of these antibodies is increasing with the passage of time, but their pathological significance is not yet well defined. As mentioned, these antibodies have been found associated with multiple rheumatic diseases, systemic sclerosis, polymyositis, dermatomyositis. There is increased prevalence of these antibodies in neonatal lupus, especially in pregnant women, rheumatoid arthritis. They have also found associated in certain tumors like lymphomas, autoimmune liver disease, and around 3% of healthy individuals may also carry anti-SSA antibodies. So basically, anti-SSA system basically comprised of two different autoantigens of around 60 and 52 calodactyl proteins. Previously, it was thought that these two antigens are different, but latest researchers figured out that initially it was found that they are similar and they are part of a big macromolecule. But later on, latest researchers identified that these two autoantigens are entirely different in terms of their structure, function, and cellular location. Like Rho60 is located in the nucleus and belongs to TRO family of proteins, which plays an important role in repairing misfolded non-coding RNAs and the cell survival followed by the exposure of ultraviolet radiation. In contrast, IL-52 is located in the cytoplasm and it is basically an E3 ligase of the TRIP family, which has a number of functions like production of pro-inflammatory cytokines, induction of apoptosis, and also provide antiviral responses. So basically, the clinical association of anti-SSA antibodies in rheumatic disorders is quite controversial. A study mentioned that around 6% of rheumatoid arthritis patients had these antibodies who developed joint involvement along with the progressive disease. Another study mentioned the association in non-erosive deforming arthropathy who had systemic lupus erythematosus. Another study mentioned the close association of antibodies in late onset SLE, which is basically a benign disease which manifests like with interstitial lung disease and neurological characteristics, but the renal flares were less commonly seen in those individuals. Barbara et al. Evaluated SLE patients and they found that anti-Rho52 and SSAB antibodies are directly implicated in the pathogenesis of lupus nephritis. So again, association of SSA antibodies in lupus nephritis is not well established. Some studies have shown no correlation, whereas other studies have mentioned that these antibodies alone are associated with high prevalence of nephritis. Arbunkel et al. Did a study in around 130 SLE patients and he found that in around 48% patients, SSA antibodies were the first antibody to identify and they were present prior to the diagnosis of SLE. Another study highlighted possible role of anti-Rho antibodies in the pathogenesis of lupus nephritis, but they didn't mention the underlying mechanism. Yoshimi et al. Did a study in SLE patients with low complement levels and they found that anti-Rho antibodies do manifest with cutaneous manifestation in polyarthritis and CNS features, but renal manifestation were lacking in this cohort of the study. So with this background information, we performed a study to look for the presence association of anti-SSA antibodies in glomerulonephritis patient. We evaluated around 200 patients of glomerulonephritis. They all were they all had anti-cell antibodies in their serum. In these patients, we look for the presence of anti-double-stranded DNA and anti-ENA. So, autoantibodies were found around 56% of these patients. Out of them, anti-double-stranded was present in 28%, whereas anti-ENA was present in 47% of these patients. In anti-ENA positive, single antigen specificity was present in 59%, whereas more than one anti-SSA antibodies were present in 41% of this cohort. As expected, anti-SSA antibody was most frequently present, around 62%, followed by other anti-ENA, anti-stones, ribosomal T protein, and many others. When anti-SSA antibodies, which were present in 58%, these antibodies were mostly present along with other anti-ENA antibodies in significant patients. Whereas in isolated anti-SSA in significant patients, like 53%, whereas isolated anti-SSA antibodies was reported in 47%. When we found the association of anti-SSA antibodies with other anti-ENA, we found that almost all SSB antibodies were found along with anti-SSA antibodies, which has been reported earlier. They both are present simultaneously. But interestingly, we found that anti-SSA antibodies were significantly associated with not only SSB antibodies but also with ribosomal protein antibodies. When we assess the renal function of these patients, around half of the patients had renal dysfunction in terms of the range serum creatinine. Evaluation of anti-cellular antibody titers showed significant association with higher titers were present in SSA antibodies. Like in around 60%, the ACA titers were either equal to or more than 1:320. When we diagnose these 58 SSA antibody patients, the most common clinical diagnosis was lupus nephritis, which we found in 88%. Next was overlap syndrome, which was detected in 10%, and only one patient had systemic sclerosis. We biopsied around 86% of these patients, and class 4 lupus was most common in this cohort of the patients. When we assessed the clinical features of these patients, we found that anti-SSA antibody patients were more significantly presented with features of oral ulcers along with photosensitivity, whereas other features of glomerulonephritis were also present, but they were not significant, like proteinuria, oliguria, hypertension, Raynaud's, and many others. Assessment of anti-double-stranded DNA and anti-SSB antibodies along with anti-SSA antibodies did not show any difference in mean serum creatinine. And this finding was in contrast to our previous published data, in which we saw that though it has been already reported that double-stranded DNA are significantly associated with lupus nephritis, but when these antibodies were present with ribosomal P protein, the serum creatinine goes down on those patients. But here we didn't find any such association. To conclude, this study significantly shows a strong association of anti-SSA antibodies with lupus nephritis. Anti-SSA antibodies, though they are quite common, but in patients with glomerulonephritis, they should alarm the nephrologist in order to rule out lupus nephritis. And if the diagnosis, the autoimmune rheumatic disorder is not present at the time of the presentation, then they should be closely monitored to prevent renal impairment, because studies have mentioned that these antibodies appear years before the actual disease development. Having said that, large and multi-center studies are required to validate these results. Thank you for your patience. Any question?
Thank you very much, Dr. Sabhat, for such an enlightening presentation. House is open for discussion.
Thank you, sir.
Thank you very much for a very enlightening presentation. Just a small query. You mentioned that in a certain number of patients, it was seen alongside SSB, and in another group of patients, it was seen alongside ribosomal P protein antibodies. So, were there any demonstrable clinical pathologic differences in these two groups as well?
Actually, majority of patients were lupus nephritis. So ribosomal P protein is strongly associated with lupus. So clinically, because the number of different pathologies is quite low. Out of 58 SSA patients, 51 were lupus nephritis. Only 7 were non-lupus. So we cannot, in such a small number, we cannot find that correlation.
I was curious if, for example, if you followed these patients for disease severity and end organ damage, like let's say 6 months and 1 year, are we going to see any differences or just a selection?
Yes, you have rightly mentioned. Because as I have mentioned, that autoantibodies actually appears years before the disease progression. So following these patients may be associated with deranged renal functions, which was not present at the initial presentation.
Thank you, Dr. Sabhat.
Thank you so very much.
Our next speaker is Dr. Wajiha Musharraf. She is Assistant Professor in the Department of Immunology, SAUT. She did her FCPS from SAUT in 2015 and has multiple publications about renal transplant. She will talk about clinical relevance of HEP2 in direct immunofluorescence autoantibody testing according to ICAP. Dr. Wajiha.
Thank you, Dr. Tiku. As you mentioned, I'm doing more of transplant now rather than ANA. But what has got me back to looking into ANA is this introduction of the ICAP and the our association of diseases with it. So autoantibodies have been hallmark of immunity since ever, and ANA testing has played an important role in the diagnosis of the autoimmune diseases. Dating back to 1948, LE cells were first described, and later in 1950s, Kuhn Plan described an immunofluorescence method for the detection of antigens in the tissue. In 58, the first time an IFA was described for the detection of ANA, and later in 1959, the LE cells function was described. Initially, ANA was detected on cryopreserved sections of rodent tissues, and it was used for many years. With the introduction of HEP2 cells in the 1970s, which were derived from the laryngeal cell carcinoma, they were thought to be better than the rodent tissue because they could be produced in large numbers. The production was easier. They had bigger nuclei, and they expressed more antigens as compared to the rodent tissue. And when produced, they were producing monolayers, so observing these cells was much easier. And IFA-SN-HEP2 is a method capable of detecting a wide spectrum of autoantibodies. A positive HEP2 ANA test is part of classification and diagnostic criteria for many autoimmune diseases such as SLE, juvenile idiopathic arthritis, autoimmune hepatitis, and primary biliary cholangitis. ANA detected by IFA is the antibodies are directed to multiple structures in the cell, which could be against the nuclear constituents, it could be directed against the nuclear envelope or the mitotic spindle apparatus, the cytosol, the organelles, or the cell membrane. And all of these give us a different clue for the disease association. IFA, according to ACR, has been always said to be the gold standard for the detection of ANA antibodies. So we have been doing ANA since ever, and we have been reporting, but it was thought that there is a need for harmonization of the terms we are using for ANA, globally for the patient, better management and for the patients. So, a committee for the standardization of autoantibodies and rheumatic and related disease, which was established in early 1980s, it came up with a community on serology, which is now known as the Autoantibody Standardization Committee. It's a subcommittee of IUIS, and the main function was to produce global standardization and harmonization of the autoantibody testing through peer-reviewed articles, through distribution of reference materials, and continuing education. The first ICAP was initiated in 2014 at Sao Paulo in Brazil, and with this, these continuous workshops and ICAP sessions kept coming up, and the idea was to generate a consensus of the patterns which were produced from the HEP2 cells. Any term is now outdated and confusing. Why is it confusing and outdated? Because when we are doing IFA on HEP2 cells, we see antibodies which are directed against many organelles of the cell, not only the nucleus. You see autoantibodies against cytoplasm, you see autoantibodies against the mitotic apparatus. So ICAP now recommends to use the term HEP2 IFA. So what has ICAP done? It has produced a nomenclature and a classification tree. And as you see here, there are four categories. The first one is the negative, which has been labeled as AC0. AC is the AC stands for anti-cell, which was given by the Brazilian Consensus Committee. And AC0 is negative. Then they are nuclear, where there are 15 IFA patterns described in the nuclear patterns, 9 in the cytoplasmic, and 5 in the mitotic. And as you see, there are orange and green. The orange ones are the one which can be reported at the competency level, and the green ones can be reported when you have experience and the expertise to report them. Nuclear patterns start from AC1 to 14, and then the last one is AC29. This is how the pictures are very nicely given in the ICAP website for the reference. Then these are the cytoplasmic patterns. It's very easy to recognize the familiar speckle, AMA, Golgi, and algebraic patterns. But to look for these, you need expertise and time to be able to report that. So this is how the cytoplasmic patterns look. These are the 5 mitotic patterns. So after these patterns were described by ICAP, what change has been brought? This is initially how we used to see a pattern. We would recognize it homogeneous or speckled or nucleolar, and then we would have a disease association. Okay, maybe it's SLE or maybe this. But now with the advance of ICAP, what we are able to do is we are able to give the relevance that it can be found in which diseases, and if certain disease is suspected, which reflex test you have to do, and if what follow-up test you have to do. And in certain diseases, they recommend if further test is required or not required. Right. And how can they be reported, and how the clinician should be informed about the patterns. Okay, so now we know that these patterns have been laid down. So how do we go about it? How can laboratories begin learning about ICAP? It's very easy. A free registration is possible at the ICAP website, and a very good training module, which is very easy to understand, is present there, which consists of the introduction to ICAP. It tells us about the technical recommendations on performing the HEP2 test, and the recommendation for dividing IFA patterns into competent and the expert levels. So why do we should we adopt this ICAP method? Because this test is being used worldwide for screening autoantibodies. The pattern informs us about the putative autoantibody specificity, and the goal of ICAP is to promote harmonization of HEP2 pattern nomenclature, refine interpretation and test result report, and emphasize immunological association and the clinical relevance of these patterns. So is it being adopted? Yes, it is. IK website has been translated into 19 languages, and there have been more than 3 lakh views per year from over 180 countries. This is what is the screening dilution which is recommended. This was a study which was carried out by most of the members of the ICAP society. 118 laboratories from over 68 countries were enrolled, and the screening titer which most of them did was 1:80. The endpoint titer, which most of the laboratories were performing, was 1:1280. Okay, so is it possible to report estimations of titers based on the fluorescence intensity and the screening dilution? One may be able to look and tell, but this is not recommended. And if you have to tell about titer, you have to have proper titrations done to be able to comment on it. And how should different patterns be reported in different dilutions? Like if you have 2 patterns being seen on the slide, you have to report them separately. For example, if you have nuclear dense fine speckle, you say AC2 and tell about the titer it is positive, and the second pattern which is positive, you have to tell that separately with its titer. There's much discussion about it, that should cytoplasmic patterns be reported as ANA negative or positive. The thing, it comes back again that the term ANA is misleading. We should be using the term as anti-cell antibodies because you're looking at all the antibodies in the cell. And yes, anti-cell antibodies will be reported as positive. Okay, and when we look at the nuclear patterns, even it took us time to understand how to differentiate between nuclear patterns which are designated as AC8, 9, and 10. It requires expertise, it requires time to understand. So people who are at the competent level, they should, if they're not able to discriminate, they can just say, okay, the pattern is AC8/9 and 10, and then clinical correlation is required. Okay, so the next question is, should we inform which secondary antibody was used in IV reactions to the clinician? I can suggest that it's not really necessary because actually the clinician is not looking, he is looking for the answer, so antibodies are not required. And then there are certain instances when you are not able to identify the actual pattern. You're not able to classify into AC1 or which AC ever. So in that case, ICAP recommends that you describe what you see in the microscope clearly. Like if you're seeing a fibrillar pattern in the cytoplasm, or you're seeing multiple shades of green, you describe that properly in your report, and you could give them a code of ACXX. Okay, so when are reflex tests important? Like most of the countries are highly dependent on IFA. They're not looking for specific test after AA positivity. The reason is mostly due to economic considerations. But in cases like if there's AC3 pattern, these are exceptions where you do not need to do reflex testing. You have a diagnosis, but in case you have AC2, you have to check for DSF-70 antibodies. Otherwise, you cannot report. You cannot give the proper information to the clinician whether it is in a normal person or is it in autoimmune disease. So at times, if autoantibodies against SSA R52, SSA R60, Jo1 or RNP are present, then they may give a negative result on the Hep2IFA. So I can suggest that you acknowledge this, that the negative would be due to this and clinical correlation is required. So how far is ICAP? It's just a smartphone away. A very easy to use app is available on smartphones, and you can download the app and you can register very easily. So actually, ICAP is a smartphone away, or maybe, yeah. So this is an article published in Autoimmune Highlights by Vancouver, and they have said that combining IFA with a specific extractable nuclear antigen antibody is more informative rather than just a simple IFA. So looking for the specific antibodies is important. This is a study published by Dr. Sabiha and her group. They have also seen the antibodies in association with the IK patterns in their setup, and they have said that getting ANA specific antibodies on ANA patterns is not really possible. So you need to go for reflex testing. So what are the perspectives of future directions? This is what has been happening, the initiation with the LE cells and then the HEP2, and now we are going towards the automation and using artificial intelligence. And image-based computer algorithms are present, ANR in clinical laboratories. It's very easy to report positive and negative on these, but the interpretations are still dependent on technical operators. The computer and the AI are very good at identifying something, but still the human brain, what it sees and how it interprets remains of utmost importance. To conclude, ANA is test seems in inappropriate terms now. Clinicians must be aware of the details on how ANA was performed in the laboratory. Sorry for the typographic error. And reports of F2IK should include autoantibody association of the specific AC pattern, suggestions for further follow-up testing, and where to find information on every aspect of the anti-cell antibodies. And when there is a clear cytoplasmic or mitotic apparatus reactivity present, the results need to be reported to the clinician. Initially, हम लोग सोचते थे कि cytoplasmic pattern है तो हम बस ANA negative करके जाने देते थे. But these cytoplasmic and mitotic apparatus are of great importance. And laboratories should focus on the training personnel and building image libraries for using guidance or assessing ANAs, and improve inter and intra-laboratory correlations and clinical classification of patients. To end my note, if clinical relevance is not taken into account, so this is what happens. Thank you. If there are any questions.
Thank you, Dr. Vijaya. Open for discussion.
Dr. Vijaya, thank you very much for a very nice presentation. As you pointed out, the development and the evolution in the classification scheme of ANA actually developed in tandem with the development of newer automated and semi-automated pattern recognition technologies as well. And we have different semi-automated and automated fluorescent detection techniques which list the number of codes that they can confidently identify and the ones which they cannot. I'm just curious, it's a small comment and inquiry. Now there's, as you mentioned, ACXX, and similarly at the level of reporting, there are two clearly identifiable levels of proficiencies, the basic competence and the expert level competence. So is it not going to be so that a number of proficiency related issues will actually end up being labeled as ACXX when we transition to this in non-automated fluorescent assay laboratories?
So actually, as we say, KNA testing, IFA testing is actually very important and has to be done by properly trained personnel. IFAs can be reported as positive at very low titers if proper dilutions are not done, everything. So yes, competency level matters. And you can exclude, but it's a long way when automation can actually take the role of what we are seeing. You to report those XX when maybe there are always flaws. Misreportings might be done, but totally depending on automation, I think still we are a lot far away from that. And probably we couldn't use pictures in the phones, but now we're doing that, so this will also happen. But we need time.
Like, I'll rephrase it for them. The predictive value of the various semi-automated and automated immunofluorescent assays in discerning the type of the pattern is not similar for all the patterns.
Yes.
Some are more easily discernible and some are not. The similar thing is going to be with the manual observer as well.
Yes.
So I don't know, maybe once we transition to this, maybe multi-center studies to follow up, if there agreement between the types of.
This transition would actually require a consensus among the, I would say, the laboratories which are maintaining standards, who have the expertise. They should have an inter-laboratory collaboration and come to a consensus and then compare to these automated machines. So
I have seen this automation done elsewhere outside Pakistan. तो वहाँ पे लोग ये करते हैं कि जो मेजर पैटर्न्स है ना, जो स्पेक्ट्रल है या होमोजेनस है, इस तरह के, they can report it easily from the images they are getting. But for other images, they do look at the हाँ, क्योंकि बहुत सारे mixed patterns भी होते हैं जो वहाँ पे हम लोग नहीं कर सकते, इसको define नहीं कर सकता हमारा automated system.
उसमें how much pattern, कितने patterns की reporting पे आप immediately switch कर सकते थे जब आपने उसको adopt किया अपने setup आप एक
कौन सा ये जो तुमने
जो आपकी स्टडी फिलोसफी जो आपकी अभी पब्लिश हुई स्टडी,
अच्छा अभी जो मेरी स्टडी तुमने कोड की थी, तुम किस की बात कर रहे हो? क्या हमने कोडिंग जो स्टार्ट किया था?
रिपोर्टिंग पैटर्न जो नए बने अडॉप्ट कर लिए।
हाँ, उसके अंदर हमने ये किया था, देखो वो एक्चुअली हम कहीं और से तो वो नहीं कर सकते थे कंपैरिजन, जो हमारा जो एरियल पैटर्न का जो वेबसाइट है ना, हम हर अपने उसको इमेज को जाके उसके ऊपर देखते थे और उससे कोरिलेट करते थे कि हाँ ये वही पैटर्न है और हम जो रिपोर्ट कर रहे हैं वो सही कर रहे हैं। एक जो चीज़ वो थी। दूसरा हमारे पास कैप के सैंपल्स आते हैं, तो जो कैप के सैंपल्स आते हैं ना, उसकी वजह से तो हम जो पैटर्न दे रहे होते हैं हमें कह रहे होते हैं कि हम सही किया। इनका आता है ड्राई चैलेंज, तो ड्राई चैलेंज इज वेरी वेरी डिफिकल्ट। तो हमारा आज तक कभी भी कोई फेल नहीं, तो हम शुरू में तो हम इस तरह से रिपोर्ट नहीं करते थे, हम लिख देते थे, मगर फिर उसके बाद हमने देखा कि हाँ हमारी इतना कॉम्पिटेंस लेवल हो गया है, नाउ आवर आईज आर नाउ रिकॉग्नाइजिंग इट। उसके बाद हमने अपना जो पैटर्न है ना वो चेंज कर दिया और अब क्लिनिशियंस को भी समझाना पड़ा। तो हमने पहले इस तरह से स्टार्ट किया था कि हम सिर्फ ए में और साइटोप्लाज्म में रिपोर्ट कर रहे थे। उसके बाद जो वो उनको आदत पड़ गई, वो शुरू में कहते थे आपने ए ने नेगेटिव लिखा है मगर आप ये पॉजिटिव दे रही हैं, तो उनको एक्सप्लेन करना पड़ता है हाँ ये ए ने नेगेटिव है, इसकी साइटोप्लाज्म में जो पॉजिटिविटी है उसकी रेलेवेंस है, उसके देखिए हमारे कमेंट्स। अब जब उसकी आदत उनको हो गई तो अब हम तीनों इसको अलग अलग कर रहे हैं, पीडी को कर रहे हैं, साइटोप्लाज्म को कर रहे हैं, माइटोटिक को कर रहे हैं। तो अब उनको बहुत ज़्यादा कोशिश नहीं होती, लेकिन नाउ अल्हम्दुलिल्लाह with continuous education and
so this is similar approach we chose, and this is how we did. Initially we started looking by ourselves, we were not reporting, then we started reporting on the same ANA के cytoplasm के नीचे कमेंट आता था, but now we have changed, हम mitotic, nuclear और cytoplasmic अलग report करें, and then people are understanding.
Last question please.
बस शॉर्ट सा जो वर्कशॉप हुआ था रोल ऑफ इम्यूनोफ्लोरेसेंस इन ऑटो डायग्नोसिस ऑफ ऑटोइम्यून डिजीजेस 15 को, वहाँ एक बच्ची ने मुझसे सवाल पूछा था कि देयर इज अ होल स्पेक्ट्रम ऑफ ऑटोइम्यून डिजीजेस, तो एक पर्टिकुलर ऑटोइम्यून डिजीज में ये वाली मतलब एंटी न्यूक्लियर इस वक्त मैं वर्ड यूज करूँगी क्यों होती है और बाकी नहीं होती है?
That's a pattern you're seeing on this slide. The disease is caused by the antibody. The binding of the antibody to specific structure of the cell is giving that pattern.
उसका सवाल ये था like if you take the example of SLE, उसमें डबल स्ट्रैंडेड डीएनए की ज्यादा धारे बनती तो सारे ही है ना, उसमें ये सवाल है जी एक में ये क्यों बनती है, जिस तरह लिमिटेड फॉर्म ऑफ एसएस में एंटी सेंट्रोमियर बनती है, उसमें बाकी क्यों नहीं बन रही? तो
like autoimmunity is confusing and
नहीं ना
मतलब इससे वो question है ना जो बचिया के हैं वो कहाँ उसकी बाइंडिंग हो रही है, उस हिसाब से पैटर्न
Thank you. Now we shall start with the free papers session. First paper will be presented by Dr. Zain from AFIP. He will talk about myelin oligodendrocyte glycoprotein antibody associated disease, clinical pathological features in Pakistani patients. Dr. Zain.
In the name of Allah, the most gracious, most merciful. Global burden of autoimmune disease is 5%. Average diagnostic delay is 5 years, and it takes at least 5 doctor visits to reach proper diagnosis. Autoimmune diseases are not recognized as urgent health issues, yet sharing major portion of morbidity and mortality. Assalamu alaikum. I am Dr. Muhammad Zain Ashraf, resident in Lahore, and today I shall be presenting my topic, myelin oligodendrocyte glycoprotein antibody associated disease, MOGAD, clinical pathological features in Pakistani patients. MOGAD is rare demyelinating antibody mediated disease of CNS. The prevalence of MOGAD in Asia is estimated to be 1.34 per 100,000 individuals. Differential diagnoses include neuromyelitis optica, spectrum disorder, and multiple sclerosis. 6 to 14% cases cause blindness. International MOGAD panel proposed diagnostic criteria, which is based on core clinical features and anti-MOG IgG antibody positivity by cell-based assay, along with exclusion of other differential diagnoses. Objective of this study was to characterize clinical pathological features of patients with MOGAD in local population. This study was conducted at Armed Forces Institute of Pathology over the period of 20 months. Study design was cross-sectional descriptive and non-probability consecutive sampling technique was used for sample size of 4. Patient was inducted as per mentioned criteria. Study included 400 suspected patients of MOGAD presented at Armed Forces Institute of Pathology for anti-MOG IgG antibody testing. Clinical pathological characterization was followed by anti-MOG IgG antibody testing. Commercial anti-MOG indirect immunofluorescence slide was selected. Patients' samples were dealt along with positive and negative controls. Fluorescence was observed using immunofluorescence microscope and observation were noted in pre-designed performer by 2 independent observers. Data was analyzed using SPSS version 23. Qualitative variable including gender, positivity, and intensity of anti-MOG antibody were expressed as frequency and percentages, while quantitative variables such as age were expressed as mean and estimate. Chi-square test was used to compare positivity in male and female. P-value of less than 0.05 was considered statistically significant. Total 400 patients were tested for anti-MOG antibodies. 154 were females and 246 were males, among which 11 patients tested positive for anti-MOG antibody, including 4 females and 7 males. Overall, 2.75% patients tested positive for anti-MOG antibodies. Among them, 64% were male and 36% were female. Result of few patients on commercial indirect immunofluorescence slide were as flashed. All patients at time of diagnosis have 2 or more core clinical characteristics. Most prevalent clinical features at time of diagnosis were optic neuritis, transverse myelitis, headache and fever, which were present in 82% patients. Common presenting characteristics among patients with MOGAD were headache and fever in 82% patients and optic neuritis in 18%. Findings of our study in comparison to other regional and international studies are presented on this slide. Average diagnostic delay in our study was 20 months, which was in coherence with finding by Nikita et al. I would like to conclude by saying that high index of clinical suspicion for core clinical features of MOGAD hold the potential to accelerate the diagnostic process, thereby mitigating the risk of morbidity and mortality. I thank you all for patient listening.
Questions, please.
Can I ask just a very basic? Probably you should have mentioned the inclusion criteria. Number 1. Number 2, how the sample size 400 was calculated. That should also be pointed out, so that younger generation is there, so that they should know how to plan these studies. And lastly, the results. Probably you got an ample time. Should have been elaborated that what was the basically key features of the results, whether they were statistically significant or not significant. They should have. Thank you.
Sir, thank you very much.
How did you calculate the sensitivity? Can you please go back to the slide? I mean, you looked at the anti-MOG antibodies in patients.
Yes,
So did you take the control population also?
Ma'am, actually, we take 400 samples which were suspected of MOGAD, and then those 400 samples were tested, and मैम बेसिकली हमने 400 सैंपल लिए उन्हीं को जिनको क्लिनिकल सस्पिक्शन थी, उन्हीं में से मैम उनकी टेस्टिंग हुई और जो क्लिनिको पैथोलॉजिकल फीचर्स थे जिनमें मॉक आईजीजी एंटीबॉडी पॉजिटिव आई थी उन्हें मैम डिटेल में कैटेगराइज किया.
Inclusion criteria, if you can mention your inclusion criteria.
Ma'am,
So what was the exclusion criteria?
Yeah, or you can mention that.
What was your objective? Sorry, can you do that again?
Ma'am, to characterize clinical pathological features in Mughal.
In Mughal in local populations, so you characterize the clinical pathological features in patients who had positive Mughal. So did you see the clinical pathological features in who were negative for Mughal then?
Ma'am, no. Only a Mughal positive patient were characterized in detail.
What was the difference?
I was just wondering that why you visualize the data in Excel and analyze it in SPSS. It was written in the slide by the method section. I suggest it should just be one software if possible.
So thank you.
Lastly, your objective was to see these alternative bodies in the local population, whereas your title reflects that in Pakistan. So we should be, all these things should be.
Sir, actually, AFIP is tertiary care center. We receive sample from all over Pakistan.
I agree, beta. लेकिन ये है कि in the study you have to justify that each part of the Pakistan is being represented. Just a suggestion.
Thank you.
Thank you very much, Dr. Zain. Our next presenter is Dr. Tajla Muhammad from Shifa International Hospital. She will talk about anti-MOG antibody associated disorders in Pakistan, a retrospective of it.
Bismillahirrahmanirrahim. I have nothing to disclose. In 1976, Lahore et al. Proposed that there was a component of myelin which was responsible for an immune response causing demyelination of the central nervous system in experimental guinea pigs. Later, Linnington et al. Then identified a mouse monoclonal antibody which was targeting a glycoprotein in the CNS in these guinea pigs, which was later identified as anti-MOG antibodies. And since these antibodies bound to MOG, the term MOG antibody-associated disease was coined. Myelin oligodendrocyte glycoprotein is a minor component of myelin. It is present on the surface of oligodendrocytes exclusively in the central nervous system. Autoantibodies associated with MOG are called MOG antibody-associated disease. It is an autoimmune inflammatory demyelinating disorder, which is typically characterized by optic neuritis, which is usually bilateral, transverse myelitis, which is typically longitudinally extensive, and acute disseminated encephalomyelitis, which usually occurs in children less than 11 years of age. These phenotypes may occur alone or in combination, and they may follow a monophasic or a relapsing course, and they may result in residual disability. Demol et al. In 2020 reported the incidence of MOGAD in Europe as 1.6 to 3.4 per million people per year. According to the proposed diagnostic criteria, all 3 points must be fulfilled where the presence of a core clinical demyelinating event is necessary, which is most commonly optic neuritis, transverse myelitis or ADEM, and less commonly cerebral monofocal polyfocal deficits, brainstem or cerebellar deficits, and cerebellar cortical encephalitis. The next criteria is a clear positive MOG IgG antibody test on a cell-based assay, and this is preferably with serum, and finally the exclusion of a better diagnosis. Here I'd like to state the importance of anti-MOG antibody IgG testing with a clear positive response. In case there is no clear positive test, then supportive clinical or MRI feature is required, along with a negative anti-aquaporin 4 IgG test. The objectives of our study was to present the clinical pathological features as well as treatment outcomes in the patients who tested positive for anti-MOG antibodies. After approval from IRB Shifa International Hospital, Shifa Tamir Millat Islamabad, all the samples which were referred for MOGAD underwent indirect immunofluorescence using EEU90 cells which were transfected with MOG genes. The inclusion criteria were all the positive patients between January 2018 to December 2022, and all the patients who had clinical details available. Exclusion criteria were the patients who did not provide consent, and data was collected via a structured questionnaire. Out of a total of 740 patients, 626 patients were negative for anti-MOG antibodies, 114 patients were positive, out of which 55 patients were excluded, as 53 patients' details were not available and 2 expired. We had 59 patients included for the final analysis. Here I want to state that the patients who were positive for anti-MOG antibodies, 81 of them also underwent testing for anti-aquaporin 4 antibodies, which were negative. And although a double positive is a rare occurrence, there have been a handful of reports across the globe. These are the tests carried out at Shifa, where A, we have a negative control for anti-MOG antibodies using EU cell line, and in B, this is a patient's positive test showing green fluorescent with a granular cytoplasmic staining with accentuation of T cell membrane. According to our study, the prevalence was 15%, mean age was 23 years with the age range of 4 to 59 years, and there was a male preponderance of 69%. The most common clinical features was a combination of vision loss, where we had 28 bilateral and 11 unilateral, along with muscle weakness and headache, followed by tingling, vomiting, and constipation. The most common phenotype was a combination of optic neuritis and transverse myelitis at 41%. Then we had transverse myelitis alone at 26%, optic neuritis alone at 25%, and ADEM at 8%. Here I want to comment that 15 patients were aged 12 and under. Out of them, 5 patients presented with ADEM and transverse myelitis. The different treatment given was a combination of first and second line immunosuppressive therapy, where the most common were oral and IV steroids, along with azathioprine being the most common modalities. Response to treatment, we had 27 patients who fully recovered. Out of these 16 patients, there was no symptom recurrence, and the treatment included a combination of IV and oral steroids, plasma exchange, and azathioprine. Then we had 49 patients with partial improvement. Out of them, 6 patients had documented relapses. Then we had 5 patients who did not improve. This was a follow-up of the patients. Here we had a 1-year follow-up of 28 patients, where 9 fully recovered and 18 showed partial improvement. Then we had a 2-year follow-up of 6 patients, where 2 showed full recovery and 3 patients showed partial improvement. A 3-year follow-up of 4 patients showed 3 showed partial improvement and 1 patient showed full recovery. A 4-year follow-up of 7 patients showed 3 each for partial recovery and full recovery, and a 5-year follow-up of 1 patient showed partial improvement. In conclusion, I would like to state that the clinical presentation for MOGAD is most typically optic neuritis, transverse myelitis, and ADEM, especially in children less than 12 years of age. These phenotypes occur alone or in combination. These affect both children and adults, and initiation of treatment is vital as there's a favorable response. Thank you very much.
Thank you very much, Dr. Khan. Any questions?
So you said that some of your patients had dual positivity.
They did not have dual positive. My patients did not have dual, but there have been a handful of case reports around the globe.
Okay, but none of your patients had dual positivity, and you have checked both antibodies in them?
Yes, in 81 patients we checked.
Okay, in 81 patients they did not have it. It was only anti-MOG.
Only anti-MOG.
So that means that we don't have the other antibodies present, or that's other?
They were not tested, so.
Okay. So this IVIG, I think, is a standard therapy for patients with these kind of autoimmune diseases.
Yes.
And you showed considerable improvement in patients. So all those patients, they will receive conventional immunosuppression or IVIG, and what was the combination?
It was some received only steroids, some received a combination of IVIG plus azathioprine plus steroids. So they all had different, according to the presentation.
In one cohort of anti-MOG antibodies, you had different kind of severity of the patient.
Yes, ma'am.
Thank you very much for an excellent presentation.
Thank you.
I was just curious, the cases where you tested for anti-aquaporin 4 and they turn out to be negative. Now, anti-aquaporin 4 associated disease, there is a distinct phenotype which is negative for anti-aquaporin 4 antibodies, but still has the neuromyelitis optica spectrum disorder. So, was your inclusion criteria sufficiently tight enough to exclude that particular group from your group of patients because they are known to have low titers of anti-MOG IgG antibodies? So that's just a suggestion that maybe you can go back to your data and try to exclude that because the percentage positivity that you're reporting is much higher than that's in literature. So I presume that some of this includes the low titer antibodies in anti-aquaporin 4 negative neuromyelitis optica spectrum disorder, which, as you pointed out, is probably, it seems to be part of the cohort. So if you segregate that totally, for example, if you just segregate all the ones where there was some suspicion of neuromyelitis optica spectrum disorder, because there was that suspicion that that's why they were being tested for anti-aquaporin 4 antibodies.
Yes.
So if you exclude that subset, then what's the positivity? So that will also be an interesting thing to see.
A small comment. During the study, you mentioned the excluding criteria. Those who did not give the consent, you said that you excluded.
Yes.
Well, this should not be the exclusion criteria. Exclusion criteria तो basically whatever you select, उसके अंदर से फिर आप reject करते हैं. For example, if you are studying diabetes, उसमें आप देखते हैं कि diabetes के patients में, for example, जिसको hypertension है, you want to exclude those patients. So it should be your inclusion criteria, and out of those, you have to exclude people, not those people who do not want to participate. Thank you.
Okay, just very nice presentation. Just to know, do you have any history of the patient, like family background and genetic, familial reasons for this disease?
As per my knowledge, there is no HLA association. Other than that, I don't know.
Okay.
Thank you very much, Dr. Tahira. Our last speaker for today is Dr. Aftab Hassan from AFIP. He will talk about clinical pathological features of Pakistani patients with anti-aquaporin 4 antibody positive neuromyelitis optica spectrum disorder, comparison in local, regional, and international studies. Dr. Aftab.
Bismillahirrahmanirrahim. In 1894, Eugene Devic coined the term neuromyelitis optica acuta, describing a syndrome of optic neuritis and acute myelitis, now commonly known as Devic's disease. Worthy chair, co-chair, and respected audience, Assalamu alaikum. I, Dr. Aftab Hassan, will be presenting my study, clinical pathological features of Pakistani patients with anti-aquaporin 4 antibody positive neuromyelitis optica spectrum disorder, a comparison with local, regional, and international studies. Neuromyelitis optica spectrum disorder is an inflammatory disease of CNS. Its reported prevalence in South Asians is approximately 3.5 per 100,000 population. Pathogenic anti-aquaporin 4 antibody are against aquaporin 4 water channels, and they are 10 times more prevalent in females as compared to males. NMOSD denotes a significantly impactful condition as 50% of the patients develop blindness or paralysis, and 33% will die within 5 years. According to international consensus diagnostic criteria for NMOSD, the patients can be broadly classified on the basis of 4 clinical characteristics and the presence of anti-aquaporin 4 antibody, as shown in this slide. Objective of this study was to characterize clinical pathological features of patients with anti-aquaporin 4 antibody positive neuromyelitis optica spectrum disorder in Pakistani population. It was a cross-sectional descriptive study conducted at Department of Immunology, Armed Forces Institute of Pathology during a time period of 18 months. Non-probability conjunctive sampling technique was employed for sample size of 369 patients. Inclusion and exclusion criteria are as flashed. Clinically suspected 369 patients of NMOSD were included. Clinicopathological characterization was followed by anti-aquaporin 4 antibody testing and results were compiled. These are core clinical features of NMOSD, and clinical suspicion was based on presence of any of these clinical features. The methodology included kit selection, commercial anti-aquaporin 4 indirect immunofluorescence slides by Euroimmun were selected. The kit verification was carried out to verify the manufacturer's claims, and then the patient samples were dealt along with positive and negative controls. Two independent observers reported the results. Data was analyzed using statistical package for social sciences variant 23, qualitative variables including gender, positivity of anti-aquaporin 4 antibody, and intensity were reported as frequency and percentages, while quantitative variables such as age were reported as mean and standard deviation. Chi-square test was used to compare the positivity in male and female patients. Total 369 suspected patients were included, out of which 168 were females and 201 were males. Out of suspected patients, 9 patients were tested to be positive, out of which 5 were females and 4 were males. Out of 369 patients, only 9 patients were positive, including 44.5% of males and 55.5% of females. The core clinical features at the time of presentation of patients are as shown. The most common clinical features were optic neuritis and acute myelitis in 77.8% of the patients, while area postrema syndrome was observed in 66.6% of the positive patients. Immunofluorescent images of negative control and positive patient specimen are as shown. Comparison of my study with local studies revealed that mean age of onset in our study was 37 years, which was in coherence with Banu et al., which reported 38.2 years to be the mean age of onset. And a diagnostic delay of 2.7 years was observed in my study, which was lower than the other two reported studies. While comparing my study with the regional studies, a high male percentage and lower female to male ratio was observed in comparison with Indian and Iranian studies. Mean diagnostic delay of 2.7 years was observed, which was twice as compared to the Iranian study. And while comparing the core clinical features, statistically significant difference was noted between the presentation of core clinical symptoms in my study and those reported by the Indian study. Most frequent core clinical characteristics in 3 different studies are shown in this slide. Most common clinical feature was optic neuritis followed by acute myelitis, while a high percentage of area postrema syndrome was noted in our set of data as compared with the Indian study. In comparison with the international studies, again, a high male percentage and low female to male ratio was observed in our data. Mean diagnostic delay of 2.7 years was noted, which was higher than the other 3 studies. And in comparison of the core clinical features, statistically significant difference was noted in the prevalence of area postrema syndrome in my study and 2 other studies as shown. Here I'll show that in comparison with the international studies, optic neuritis was the most frequent core clinical feature with which the patients presented, followed by acute myelitis, and area postrema was higher in our group of patients as compared with the other studies. In the end, I will conclude by saying that high index of clinical suspicion in patients presenting with core clinical features of NMOSD can expedite diagnosis, potentially averting morbidity and mortality. I thank you all for your patient listening.
Any questions?
How does it affect the mortality and morbidity that we have concluded? It affects the morbidity and mortality. How does it affect?
Ma'am, basically the earlier diagnosis can ultimately reduce the complications, and the treatment can be initiated as early as the diagnosis is made. So, it can significantly reduce the ongoing complications which the patients face.
Yes, but then it should have been shown in the results, no? Summarize that and put the results.
Right.
This marks the end of the session. I thank you all for your patient listening, and thank you all the speakers for their enlightening talks. Now I would like to invite Professor Dr. Ghazala Jafri to please come on stage and present the shield and certificate to the chair first, Dr. Nadeem Afzal. Ma'am Ghazala, thank you very much.
Great.
Now I would request Dr. Nadeem to please give away rest of the certificates and sheets, please. First to Colonel Hamid Nawaz Tipu. Dr. Sabahat, please come on stage and receive your shield and certificate. Next, Dr. Vijaya, please come and receive your certificate and shield. Dr. Zain, please come and receive your shield and certificate. Dr. Taira. Dr. Aftab, please come on stage.
And Dr. Sarawakar for moderating this session. Thank you.
I would like for you closing remarks of the session, if you would like to, please.
Thank you very much. Jabardast, full of information. बहुत सी चीजें मुझे पता नहीं. It's always good to be with young people, young researchers. Just would like to share with Dr. Sabahat's contribution. I spent about 5 years in Malaysia. तो वहाँ पे भी एक केस ऐसे ही था मेडिसिन में जब डॉक्टर को देख रही थी तो सारा क्राइटेरिया उनका फिट हो रहा था एसएल में लेकिन जब हमारे पास लेबोरेटरी टेस्टिंग के लिए आता था तो उसके टेस्ट नेगेटिव लेकिन फिर उन्होंने कहा कि इसका ईएनए प्रोफाइल करते थे तो इट वाज वेरी सरप्राइजिंग फॉर मी एट दैट टाइम इट्स वे बैक अबाउट मोर देन 12 years ago. So that was positive. तो फिर हमने एक केस रिपोर्ट जो है वो पब्लिश किया. Very interesting कि उसके बाद डॉक्टर बजिया का था. Can't say much about it because it was talk in itself. बहुत सारी information थी इसके अंदर and she updated us about criteria of documenting the immunofluorescence. उसके बाद पेपर्स से भी बच्चों के बड़े अच्छे पेपर्स थे. Very interesting. उसमें छोटी-छोटी मेरी टाइम एंड अगेंस्ट क्वेश्चंस आ जाती हैं शायद probably because of my age I can't resist चीज़ों पे कमेंट करना लेकिन पर्पस ये होता है कि किसी को डिस्करेज करना नहीं होता हर्ट करना नहीं होता क्योंकि वी आर ऑल लर्निंग मैं खुद अभी रिसर्च की एबीसी सीख रहा हूँ और बच्चों से सीखता हूँ क्योंकि बच्चों का नॉलेज ज़्यादा होता है वो पढ़ के आते हैं डिस्कस करते हैं तो पर्पस यही होता है ऐसे प्लेटफॉर्म पे कि हम चीज़ों को प्रेजेंट करें और जितने लोग बैठे हुए होते हैं दे आर फ्रॉम द डिफरेंट बैकग्राउंड्स So, milk basically a learning process. All in all, it was very interesting. There was much to learn about it. Thank you very much. I would like to close this session. Thank you. It's basically all the people who worked very hard for the organization of this conference. They really deserve big congratulations. Dr. Surya, Jonathan, and there are so many other people. Thank you very much.
So there are some award to be distributed among our first, second, third winners for oral and poster presenters. So, oral posters.
For poster presentation, our third award goes to Rafia Khalid or Afshar Khalid, if any of them is here. They presented genetic polymorphisms of CCR2 gene and expression in newly diagnosed HIV patients and healthy controls. है यहाँ पे राफिया या अफशर? UHS से, they are from UHS. आप please doctor please. Sir.
Sir.
The second award goes to Mishal Shaheen. I think she's from Alpha. Sanjay, please. She presented a poster, its immunogenes, immune response varies in a VDR genotype. The first prize goes to Dr. Mohammad Zain Ashar for presenting clinical pathological features of patients with anti-NMDA receptor encephalitis experienced from a tertiary care center in Pakistan. Now the overall abstracts prizes.
एक बात बता दूं कि आप लोग की इस बार माशाल्लाह से बहुत अच्छी प्रेजेंटेशन थी। इट वाज डिफिकल्ट कि हम थोड़ा सा उसको मुश्किल हो रहा था कि हम क्या करें, मगर थोड़े थोड़े नंबर से फर्स्ट सेकंड थर्ड आए हैं। तो ओके, सो द थर्ड प्रेजेंटेशन गोज टू डॉक्टर खुर्शीद। Dr. Khurshid he actually presented on CD4 and CD17 cells in patients. Congratulations. Second award goes to Nida Javed. She's from Lahore Hospital. And first award goes to Dr. Hafza Faruqi. Hafza Faruqi is from which institute? DUHS. Okay, Sababuti. So that's all from me. Thank you very much, everyone of you. Thank you so much.